The Life Sciences DNA podcast is sponsored by Agilicium Labs, a collaborative space where Agilicium works with its clients to co-develop and incubate POCs, products, and solutions. To learn how Agilicium Labs can use the power of its generative AI for life sciences analytics, visit them at labs.agilicium.com. Amar, we've got Markus Gershater on the show today. For listeners not familiar with him, who is he?
Markus is the Chief Scientific Officer and co-founder of Synthace. He was a research associate in synthetic biology at University College London and a biotransformation scientist at Novacta Biosystems. He has a PhD in plant biochemistry from Durham University.
And what is Synthace? Where do they fit into this world of AI in the life sciences?
Synthace has created a digital experiment platform for research and development. That's a cloud-based platform that allows scientists to use a natural language interface to use AI to design complex experiments, automate their running, and capture and analyze data all in an automated way. It has integrated AI into this platform to accelerate the discovery and development of new therapeutics.
Well, before we begin, I want to remind our audience that if they want to stay up on the latest episodes of the Life Sciences DNA podcast, they should hit the subscribe button. If you enjoy this content, please hit the like button and let us know your thoughts in the comments section. If you want to listen to the show on the go, you can find an audio-only version on major podcast platforms. And with that, let's welcome Markus to the show.
Markus, thanks for joining us today. We're going to talk today about using AI to transform R&D, Synthace, and its digital experiment platform. But before we do that, I wanted to start with a piece you recently wrote that likens AI in drug discovery to the electrification of factories and time it took to realize productivity increases. Right. So tell us a bit more about it, and where do you think we are in the ultimate impact AI will have on drug discovery and development?
Wow, okay. That's quite a lot. Well, first of all, thanks very much for inviting me on. It's a real pleasure to be here. The analogy with electrification is really that when electricity first came along to be available to run factories, right, then what it was replacing was huge steam engines, which would then have these massive drive shafts that would run through the factory, and everything would be run in pulleys from that drive shaft, right? Mm-hmm. And so what that would mean is either the whole factory's on or the whole factory's off, right? All the machines are all running, or they're not running at all. And similarly, you have to make sure all your machines are arranged around that drive shaft. You have no other way of getting power to those machines. So that's what you have to do. And so when people first, had access to electricity, then some factories, they upgraded to, electrical engines instead. But they didn't actually see any productivity improvements from it, right? They'd switched power sources, but they hadn't seen any kind of productivity improvements. But we think of electrification as being kind of a revolution in the way that manufacturing was done. But it was only when people started to actually realize the benefits of electrification and reorganize their factories around it that they started to get the benefit. So essentially, it meant that now you can have any machine anywhere and you can turn them on and off at will. So now you can rearrange your factory from being centered around the drive shaft to being centered around the production process. And so then it becomes like the production line method of actually producing things, which is a hell of a lot more efficient. And so that's when you end up with the efficiency gains from electrification. So it wasn't actually electrification itself, it's the way that it could then change the way of working. But that benefit actually came substantially later than the original introduction of electrification. I think it's an interesting thing to think about because, we have this new powerful tool in artificial intelligence and it's being applied in various different ways to different parts of the value chain. But I feel like until it's actually adopted across the value chain, right, then we're not going to see the full benefit of it. So that's kind of where that analogy comes from. And I mean, the other thing that I sometimes get a bit irritated by is kind of just this generic use of the term AI, right? And I think those of us who've been, talking about AI for a while and maybe, have a bit more experience about where it can be applied, it's similarly like sort of exhorting people, oh, you should just use electricity more. It's like, well, actually, you could use electricity for all sorts of different things and in all sorts of different ways. And I think like sometimes the specificity of what computational power can do gets lost in this kind of term of artificial intelligence, which it can be helpful in that it can capture some of our aspirations for what it might do for the field, but it can also be unhelpful, right? Because it kind of obfuscates everything.
Okay. And so when you're talking about AI, right, and it's always an interesting one, like what do people mean by AI, right? So it can be like, automation with business rules to machine learning, to deep learning, to generative AI, right? So like how do you think about AI?
I tend to try and demystify it. So there's I think she's kind of a sociologist stroke ethicist here in the UK called Hilary Sutcliffe. And she said something which really resonated with me once. If whenever the term AI is used, you just replace it in your head for the word software, right? Then I think you often get to a much better kind of intrinsic understanding of what it is, because AI sometimes has this sort of mysticism or maybe like this sort of sense of the unknowable around it, right? Whereas if you say software can now enable voice recognition and like text to speech or software can now rapidly generate condensed bits of information from lots of different, then it kind of just makes it a little bit more, it makes it more tangible and more real to me somehow. And so, I think essentially it's building sophisticated software models, right, which can then help us to make predictions or help us in some way with the complexity of what we're dealing with. I think that, thinking about it that way could then be, it's more obvious then that it's incredibly diverse, right? Because software is incredibly diverse, right? So it's just a kind of mental game that I sometimes like to play to try and reframe the way we think about it a little bit more, right?
Absolutely. And another interesting thing is that so electrification is a great analogy. I've also seen a lot of times the people saying data is the new oil, right? Like the oil of the 21st century. So what are your thoughts about that? Like, which analogy do you think is better?
Well, I mean, I like the emphasis on data because I think that, again, a lot of people who've been in the space for a bit realize that actually it's more about the data than maybe the algorithms, right? If you have a high quality data set that you're training your models on, then that's a hell of a lot more likely to give you decent results than if you're I saw it referred to as dumpster diving the other day, right? You're just basically going through and just trying to find whatever you can and you'll shove it all into a model and expect it to magically come up with something. And again, maybe if we thought of it as like, okay, we're giving our software some data, then you don't kind of think it's going to do something magic somehow, right? Sometimes I think the perception of AI is that it should somehow give us some kind of magical abilities. And to be fair, I mean, when all of us started using LLMs, then they feel pretty freakily capable, right? And it's only when you start using them more do you realize their limitations and kind of, okay, yes, it's a very powerful and exceptionally compelling piece of software, but it's still software, right? But yeah, I think the whole data is the new oil thing. Absolutely. I think fundamentally it is all about the data, right? And obviously how we manage that data, how we bring it together, and then how we train very purposeful models on top of that, right? Again, I think the data have to be designed in such a way, and the experiments that produce the data have to be designed in such a way that from the outset we are thinking of the models that we're trying to build, right? And the problems that we're trying to solve. That's my personal take on it. I think you can look through retrospective data and that kind of thing, but often there's a real issue that they haven't been built for the purpose of finding out specific things, right?
Yeah. And that's been a big problem, right? A lot of times when doing data analytics, the data scientists are brought in a bit later when the data is already generated and we're just lying around somewhere, right? And then people are, okay, well, here's the data. Was it generated for this purpose? Not really, but yeah. Can you use it?
Right.
Right.
Right. And actually it reminds me of a quote from a statistician from like the 1930s, a guy called Ronald Fisher. And he said, if you call in a statistician after you've run the experiment, then the most you could ask of him is to run a post-mortem, right? It's kind of, and I think it's a similar kind of thing. Like, and I think what's encouraging these days, though, is that actually we're seeing a lot closer interaction between data scientists and wet lab scientists, to call it crudely, and so I think that gap is narrowing, and I think we're all learning how to speak each other's languages and understand each other's domain more, which for me is part of the fascination of it, right? I've been very lucky to work on the boundaries of disciplines throughout my whole career, and I've always found that the most interesting perspectives and questions come from like the different mindsets that you have in different disciplines, as well as obviously the different expertise.
Mm-hmm. Absolutely. So where do you think we are in this ultimate impact AI will have on drug discovery and development? I
think we've made a really exciting start, right? That would be my take. But like another thing that I'm really privileged about is that, in my role at Synthace, I get to go and talk to scientists from all sorts of different companies, I guess, much like, you getting to talk to them on this podcast. And what I found, and I'd be interested to hear if you've seen something similar, is that the wet lab scientists we're talking to, and they're in kind of early discovery functions, particularly sort of in target validation or assay development, those kind of areas. And they're saying, look, we're just getting slammed, right? And the reason they're getting slammed is because the bit upstream of them has been unbottlenecked by AI, right? So all this is certainly what they ascribe it to. So target identification is no longer the really painstaking academic process of like fundamental research into disease, which then as a byproduct then allows us to identify targets. Now it's an industrialized process where absolutely, and I'm sure you'll be very aware of this, of these massive multi-omic data sets, and plus other data from other areas coming together such that then these AI models are identifying the patterns in these data sets in order to then identify new putative targets. And so what that means is that, there's this flood of new potential targets. And at the same time, there's another technological shift, nothing to do with AI, but to do with new modalities coming through, like PROTAC modalities and nucleic acid modalities, which means that you no longer have to look for the typical features that you previously have looked for to look for a druggable target, right? Like a nice kind of active site that you can have a drug go and bind within. These new modalities mean that pretty much anything could be a target. So we've got a lot more targets coming in from AI, and the diversity of what could be a target has also expanded. And so what you then find is that then these folks who have to then generate the tooling for any new therapeutic program, so the assays or the cell lines or whatever, these guys now are finding that their job has just got dramatically harder, but they don't have any more resource to do it. And they're like, they've got this pressure on them to get as many new therapeutic programs started as possible. So, we kind of have called this like the new bottleneck, right? Whereas maybe target ID used to be the bottleneck. And so, there'd be a Nature paper published after, like 10 years of painstaking academic research or whatever. And then, that'd be the starting gun, and every pharma company would then race to drug that target, right? And that's the dynamic that the value chain and the process is all set up around. So it's like, fewer very high quality targets, which then everyone races to be first, right? And now the dynamic seems to have shifted quite a lot, and I think there's a good case to be made for that's happened because of multi-omics and AI models built upon those multi-omics. And so I think that's one example where the electrification has occurred, right? The electrification of the factory, but we haven't worked out how to rearrange the factory to make use of that, right? That would be my argument.
Okay, great. And so as And as you're thinking about this, right, like the drug discovery and target identification, you explained a lot, but like also thinking more about like from like the target validation or hit
Right.
identification, like how do
Right.
you see AI changing those areas?
So target validation is obviously done in a number of different ways, but I mean, fundamentally, you need to be getting in the lab, right?
Hmm.
And for a lot of these things, you could do a lot of theoretical validation of, oh yes, it makes sense,
Sure.
like which could all be done up front. But then, the kind of CRISPR-based studies, to look at a knockout of those targets, obviously is very common these days. But also like making sure that you're working with the most relevant cell lines that you can, for doing those kind of experiments, I think is absolutely vital. And similarly, kind of in assay development, I think it's somewhere where I think we need to think very carefully about the way that we're generating data going forward, right?
Hmm.
And because if we're going to build AI models on top of these, then any biases in the data, any poor quality in the data, is then going to lead to those AI models either just being ineffective or even worse, misleading, right?
Hmm.
Um, so I think that there's an application of AI still to be done. I think in the process of generating, say, exceptionally high quality assays, right?
Okay.
For example, because that's the way we produce data is through assays. So ideally, they are of very high quality, high signal to noise, but also on systems that at least some of those assays have to be on systems that are as relevant to the patient, as relevant to the clinic as possible, right?
Hmm.
And so what we're talking about now is a very high demand on these groups that I've already have outlined are pretty slammed, right?
Hmm.
And then, I mean, you mentioned hit identification as well, and I think alongside target ID, this has to be an area where AI has clearly had an impact, right?
Hmm. Hmm.
But I still think that we could be doing still substantially better were we to increase the acuity
Hmm.
and reliability of the assays that we're using throughout drug discovery as well.
Hmm.
So I've spoken with a number of people in the field, and those who are looking to up the quality of drug discovery in general tend to point at one of two things. So one of them is about the clinical relevance of the models that we're using in discovery. So
Hmm.
at Synthace, we're entirely about lab work, right?
Hmm. Yeah.
And the generation of high dimensional data sets from those lab work, that lab work, that's really what we're focused on. And the way we're thinking about then the application of these technologies is that we want to see that lab work, increasing the quality of what can come out of things. And like what I've heard is that actually often the metrics that are used for what is a good assay need to be raised.
Okay.
And the reason for this is because if you're running a high throughput screen, for
Hmm.
example, so these are the data that you're then going to end up feeding into your AI models about what chemistry could affect this particular target, right? So it's
Yep.
critical that we
Yeah.
get the best data we possibly can in that high throughput screen.
Yeah.
But
And just for the audience though, like if you can explain what's a high throughput screen, that would be great.
okay, of course, yeah. So a high throughput screen is essentially where we take, say 1 million different molecules, and we screen to see which one of those molecules can interact effectively with the target that we believe is if we that if we can hit that target effectively, then it's going to have some therapeutic effect on a disease, right?
Hmm.
So I've been talking about target ID. I realize I didn't explain that either, but so this is the process by which they identify those molecules within the cell, those proteins within the cell that if you hit them, then maybe you can maybe you can treat a disease. And what we're doing in a screen is we're running like 1 million assays with 1 million different compounds and seeing which of those compounds might affect that target. So, and what you hope to get from that is ideally lots of different compounds that might affect that target, because then that gives you some information about the kind of chemistries which might affect that target effectively. Now, the issue is that if your assay is not sensitive enough, then you're going to get a huge amount of false negatives, right? So, you've got all of your compounds and you have a threshold that you need to, any target has to get up, any compound has to get above, so you can actually see that compound has had an effect on your target, right? But if you've got a poor assay, then the threshold is higher, and it means that you've missed that compound. So then you've missed the opportunity to learn, or your AI model ultimately has missed the opportunity to learn about the chemistries that can affect a particular target. And so there are particular kind of metrics that we have against assays. One of the classic ones is Z prime, right? And Z prime is basically like a very stringent signal to noise measure. And it's measured from zero. Well, actually, it can go below zero, but like the highest Z prime you can possibly have is 1. And that's basically where you'd have a perfect assay with beautiful signal and no noise whatsoever. So you never have an assay of 1, right? But in general, it's regarded that an assay with 0.5 or above is a very good assay. But then, I've been having some conversations with a chap called Dan Thomas, who spent a large amount of his career in this early discovery space. And he makes the point that actually a Z prime of 0.5, you still got this huge amount of false negatives. And the point he makes is that false positives we can deal with, right? Because you can go back and retest them with other assays or with the same assay, and you can realize, oh, that's actually a false positive. But a false negative is lost forever, right? You'll never be able to go back, right? So it all speaks to the criticality of having very, very effective data production, right? And then we need to look at those processes, the assays, which make those data, and think about how we can make them as effective as possible, whilst at the same time trying to alleviate this bottleneck, right? So make it faster, but also better.
Sure.
And this is where, so I've outlined the problem, right? And one thing that we found really exciting at Synthace is that if you run sort of experiments which are systematically looking at all the variables that might affect an assay, but you look at them all simultaneously. So you set up this experimental design, which very systematically puts a lot of different design points through all of the different combinations about how you might set up that assay in a very systematic way. So you generate this exceptionally deliberate multi-dimensional data set, right, which describes the behavior of that assay as you move through this very high dimensional space of all the different parameters that you might change to affect that assay.
So let's do a deeper dive in that, right? So I like with an example. So first of all, for those who don't know, assays, like layman terms, experiment, right? So there's an experiment that's being done, right? So in Synthace, you have built this digital experimental platform, right? So can you take us through? Okay, well, an example where there's an experiment that's being done, and what are the challenges right now, and how Synthace, the digital experiment platform, how that is coming in and solving those challenges.
Right. So let's take the example of an assay that we want to develop, right? So this is essentially where we have to set up a system such that when a molecule interacts with a particular protein, then we get a very clear signal that it's done so.
Yes.
That's the and that signal can be measured through fluorescence, or so we have plate readers in the lab that are very good at measuring how much light is given out by a particular well. And this is often how these assays work. You have to somehow convert this interaction into some kind of fluorescent signal. That's often the job.
Yeah.
And the issue is that, if you look at that system, there can be dozens of different things that could affect that system. So like very simple ones would be things like temperature and pH, but then you also have like salt concentration and the salt type that you have in there. It could be sodium chloride, it could be magnesium chloride. You have things called reducing agents, right? So these are things that stop oxygen from degrading the system too badly, and you might have to choose both which sort you might want to use, but also the concentration of it. And then sometimes we might want to add some other protein into the system to make it more similar to the kind of crowded protein environment that you have in a cell. So there's just, and you can go on, right? There's a huge amount of factors, and often, especially with new targets, which people aren't used to working with anymore. It used to be that actually kinases were so common, right? So as a target. So then, an assay development scientist might, be tasked with developing their, the 14th kinase assay of their career, right? So they have some kind of idea about how they're going to go about it. But if you're now presented with a target, which you just have no idea of, you have no precedent with working with that kind of target, then ideally you'd be able to map out how all of these different factors, how all these different parameters might come together to affect that system, and so allow you to pick the best possible set of conditions for running that assay.
And now, so the fundamental problem here is that because of the biology, right? So in physics or chemistry, the number of factors affecting the experiment might be much smaller if you have like this controlled environment. But but in biology, it's just in a biological system is so complicated that there are just so many of these factors that need to be just right, and then as you're saying, right, like there are 20,000 proteins, right? So potentially 20,000 targets, or even like 30,000 targets that we have. We just don't know what is the right biological system in which we can get the signal, right? And that's why you have to there's this whole slew of factors that you need to consider and figure out like what is the right level for each of these factors, right?
Right. Now, but then if we think about how we were taught to do science at school, right, then you start to be taught about like the scientific method, right? And so, you're taught about the rigor of holding everything constant except for the thing you're investigating, right? And this is known as one factor at a time, right? So you very carefully just vary one thing at a time so that you can isolate the effect of that thing when compared with all of the other factors that you might be looking at, all the other parameters. Now, that's pretty good for like school level science, but I'm always a bit dismayed that the education often doesn't seem to progress beyond that, right? Actually it's perfectly possible to look at many different parameters at once, so long as you do it in a very structured experimental design. And it's particularly critical, and you alluded to this, it's particularly critical for biology because biology is an emergent system. Right, it has emerged from evolution, and so that means that it has absolutely no obligation to be understandable or simple. Right, it hasn't been designed from individual component parts that fit together neatly. It's full of unexpected interactions of these parts coming together in unexpected ways to then generate a particular phenotype, a particular phenomenon. Right, and so. And the problem with just looking at one factor at a time is you'll never then see how one factor might interact with another one, because you're looking at them in independent experiments in isolation. Whereas if you look at them all simultaneously in these very structured designs, then you can actually pick apart the way that these different components are coming together to actually generate the phenomenon that we're looking at. In this case, the assay that we're working with, right? And so. And in our experience, it's incredibly rare to come across a biological system that doesn't have these interactions all over the place, right? So where the optimum level for one parameter depends on the level of another parameter, the setting of another parameter. There's one kind of obvious way of describing what these interactions, and that's a synergy. Right, so synergy is a word that's overused, but a true synergy, like 1 + 1 = 5 or whatever, right? That is basically where you have two things coming together to give an unexpectedly good result. And in experimental design terminology, we'd call that a two-factor interaction, right? So two things coming together and we see them all over the place. And what we found at Synthace is that if you run experiments in this much more powerful multifactorial way, often referred to as design of experiments, then you can cut through biological complexity a lot more effectively. So that's like the first thing. And then if you add automation to that, then the number of runs you can do and the complexity of the design space that you can look at goes up substantially, right? So you've got these two things coming together. Like, let's look at things in a high dimensional way, but let's look at it in automated way as well. But then those experiments get incredibly difficult to plan and run, right? And this is where Synthace comes in, the digital experiment platform. Because what it allows scientists to do is say, I want to run this experimental design, and then Synthace will then work out the huge amount of intricate details that are required to actually then run that experimental design in the lab. Imagine a plate, like of this kind of footprint. So it's a typical kind of footprint of a plate in the lab, and you have 384 wells in it, or even 1536 wells. And we want to run an experiment where each one of those wells, it has a different mixture of components in there, where you could have 20 different components, right? And you're varying these all, and so no two wells are the same, right? And you're spreading all of these different experimental runs across the high dimensional design space. That is an experiment which is, to all practicalities, impossible to run by hand, and is also incredibly arduous to plan and program automation to do, right? And that's essentially the problem that Synthace is solving. So then to loop back to AI, like my hope for AI is that then, you could use these exceptionally high quality, comprehensive, multi-dimensional data sets, right? And because, what we found is that for each individual new assay, to keep on this particular example, for each new assay developed, these multi-dimensional experiments are very powerful for doing that, right? And it means that you could get to a new assay way faster, so up to 70% faster. So this is a process that typically takes up to 9 months. And we've got case studies over and over again, different clients, big pharma clients, where they've reduced that time by up to 70%. So both AstraZeneca and GSK are public that they do this kind of work with us. But also critically, we see improvements in Z prime, right? Because you're getting such a comprehensive view of that space, of that design space, you can pick those conditions which are fully optimal, right? So you're squeezing the absolute most you can out of that assay system. And then incidentally, it can often be cheaper as well, because you can find the place where it's most effective, but also uses the least of expensive reagents, because you've mapped out the space so comprehensively. So that's all brilliant, right? Each new assay, we can make it happen a hell of a lot faster and make better assays. But then what I then ask myself is, what could we do to properly unbottleneck this using AI? Is there some kind of meta-analysis we could do? Like taking, so what could the first 12 very high dimensional comprehensive data sets tell us about the 13th assay development we need to run? Or the first 50 tell us about the 51st, right? There has to be like additional information about the very nature of how these kind of systems work, which I think artificial intelligence could be exceptionally good at doing that meta-analysis, right? And so mean that we can design ever better experiments and get to ever better assays ever faster, because we're starting from a better place each time. And in that way, we've been very successful at applying AI to spaces where you can generate very large amounts of data, and you can do that with relatively kind of routine processes where you set up an automated system, then you crank the handle, right? So like it's so things like multi-omics, right? You set up your particular genomic workflow or transcriptomic workflow, and then you get a huge number of different patient samples, and you push it all through, right? And you get a huge data set which you can then apply AI to. Where we've been where we haven't applied AI yet is to kind of those areas of R&D that are a lot more variable, right? Where you never do the same experiment twice, right? Where you can't just make it high throughput in an easy kind of manner. And I think this is a real opportunity for where we can go.
Okay.
To unbottleneck this new bottleneck of target validation and assay development, but then also then push through the rest of the value chain, right?
Okay. So let's say within an assay, right? Let's say there are like 5 variables, and each variable has, let's say, 10 different values, right? And so,
Okay.
then you're talking about 10 times 10 times 10 times 10 times 10, right? Which is like,
Right.
10 to the 5, right? So
Yep.
that's 10,000, 100,000, right? So
Right.
how, like, as you're like, so the Synthace platform, how does it deal with that? Like, so like, and is, as you said, like, uses AI. So is it like looking at, okay, well, doing experiments, let's say in a couple of levels, like for a factor, then kind of determining this makes sense, then going in that direction? Like, is it making, so like, how is the AI working there?
Right. So, I mean, the process that I described for sort of multidimensional experimentation isn't actually AI. This is statistics, right? And it's a branch of statistics called optimal experimental design, or design of experiments. It's often known as DOE. Well, at least that's what it's all rooted in. And the idea there is that you set up these systematic frameworks within multidimensional space, and then. But you ask very specific questions for each experiment. And so by doing that, you can strip down the number of experimental runs that you have to do. So I'll try and give an example. So if we were to look at 6 factors simultaneously, right, then and we say, actually, to start off with, we don't know which of these 6 factors is important or not. Right. So we're not going to try and find the optimum straight away. All we're going to do is find out which factors are important. Then if you're setting up an experiment, then to see whether a factor is having an effect, if you choose your levels carefully that you set that factor to, then you can get away with two levels, right? For that factor. So you can condense the problem in that way. But then also, we have to ask ourselves, okay, so we know that there are interactions in biology, right? Like these factors could interact in unexpected ways. But what we tend to find is that the higher level that interaction, the less likely it is to occur. So I've already described what a 2-factor interaction is, right? A synergy is an example of it, right? Now, a 3-factor interaction is where the nature of that 2-factor interaction changes when you change a 3rd factor, right? So you have the interlinking, this interweaving of 3 factors together. And you can keep going up, right? 4-factor interactions, 5-factor interactions, and it just gets ever more complex. But they also get vanishingly more rare, right? So, I mean, we at Synthace have run a lot of experimental designs ourselves, and our clients have as well. And we've never seen anything more than a 4-factor interaction in the designs that we've done. Okay. In the kind of systems that we've been exploring. And so then you can make assumptions. You can say, okay, we assume that actually the most likely interaction is going to be a 2-factor interaction. And so, we're going to do a design where we strip that design down so that it isn't necessarily looking for the most complex interaction we might see, in this case, a 6-factor interaction. We know that's not going to happen. So you can make that assumption. But like, if we think about this, like it's essentially like a 6-dimensional hypercube, right? Just to, like the design that we're talking about here. So if you did every design point on the corners of that cube, then it would be 2 to the 6, right? There's a lot of runs. But that would also be looking for 6-factor interactions, which we know are unlikely to occur. So what you could do is you could very systematically and symmetrically take away design points. Until you get down to, say, I don't know, a 32-run design, right? And now a 32-run design is perfectly manageable. And the interesting thing about it is, if you imagine any one of those factors, we're actually comparing 16 data points with another 16 data points. Like, there are other factors that are changing simultaneously, but for each factor taken in isolation, you have a lot of replication around that particular factor. And so you actually end up with a lot of statistical power for then seeing whether these factors are having an effect, even as you're running these very complex experiments. Sorry, it's quite hard to explain all this.
I think this is great.
Waving my hands around, but hopefully I paint a reasonably good picture. So after you've then after you've seen which factors are important, you can then spend more experimental power on exploring exactly the effect of those factors as you optimize them, right? And you iterate your way through the design space, right? You work your way through until you find an optimum, or at least that's the classic way of doing DOE. What's really interesting in a space like assay development is they're not restricted to 32 runs. You know, their final system, they want to end up with something that works in 1536 well plates. So that means that, you know, all of a sudden you've got thousands of runs you can work with. And so then that expands the scope of these experiments dramatically. And honestly, nobody really knows what to do with all those runs, right? It's a really exciting time because, you know, in terms of like how we can experiment in the most powerful and effective way to address biological complexity. You know, like because automation and software like Synthace has now unlocked a scale and complexity of experiment which was previously impossible, like, you know, the experimental design theorists, you know, they've not caught up, right? So it's really cool. And I think, you know, this is then where there's going to be that intersection between, you know, multidimensional experimental design, automation, software, and then AI, and what, you know, like, what a fabulous data set to be training AI against. You know, if you imagine, you know, 50 different assays where you've investigated, you know, 15 factors for each of them and spread, you know, 1536 at least design points for all of those 15 factors. You know, you've got these sort of big kind of multi-dimensional data sets in each case. And often actually you don't even need to iterate these days because there is such a large number of experimental runs. You can often get to the answer just in a single experiment. So it can compress the timelines quite dramatically as well.
Yeah.
So there's a lot of huge potential. Yeah, I mean we've seen exciting things, but like what I'm always the sort of person who's then looking at, okay, so what's next? And I think, you know, there is a lot of quite justifiable excitement around AI. You know, like it is clearly an exceptionally powerful method, and we all know that it's going to go through more hype cycles, right?
Yes.
You know, like AI's been through a lot of hype cycles. It's going to go through more, but we also know that there is the heart of something very real here, right? And so it's very exciting to think about how that could be applied, you know, when you combine it with these other technologies that can produce the data sets that we're talking about, right?
Okay. And you mentioned about the Synthace system using AI for experiment for like the meta analysis, etc.
That's where we'd like to get to. You know, like, you know, at the moment, you know, like what our customers are using it for is generating these high dimensional data sets, making assays or other biological systems. You know, often like you know, cell differentiation is another area, right? You know, generating stem cells and then differentiating them into particular tissue types, you know, organoid types, all that kind of thing. That's also the very complex areas of, you know, where you're optimizing these media with all the right growth factors and things to make to make cells dance to your tune, right? You know, very, very complex and noisy systems. And so our customers are using Synthace for kind of supercharging those wet lab experiments. But I guess my excitement is that, you know, it's all about producing those data sets, right? And as we established before, like, data is the foundation for AI, right?
Yes.
And so the ultimate solution is going to be the production of these really comprehensive high dimensional data sets and then the application of AI to those data sets, right? That's my belief.
Yes, absolutely. So what is Synthace's business model and how do you work with the customers?
Sure. We have this, you know, very powerful software platform that can enable people to do these experiments and you know, get much more definitive answers from their experiments a lot quicker. And we license that on a yearly basis, like a lot of software is these days. So it's a yearly license. But what we often find is that it isn't just the software by itself. Right. So we also have a very deep expertise in these kind of ways of doing experiments within the company. We have a very long history of applying these things to biology and understanding, again, that interdisciplinary interface between the mathematics and the experimental design and what's theoretically the best designs you could run. But also then we really understand what's actually possible in the lab. and what's within the constraints of actually running experiments. So we've got a lot of expertise in that area, and so we work very closely with our clients to support them on this journey of changing the way they're doing their science. Right? It's actually quite a profound change that it ends up being, because you're going from a system where people are doing the science the way that they've been taught for however long is the correct way of doing science, to then a much more kind of high dimensional and much more powerful way. But that takes some change management. It takes it takes learning about this new way of doing things, and then obviously the software helps them to actually run these experiments so they see the results, and then that's the moment that people get converted, right? It's like, oh wow, suddenly we don't have it's such a normal experience as a biologist to get results which are a bit unclear, which are a bit ambiguous, which we're not quite sure what they mean. And then you'll have lab meetings where everybody's debating about what these results mean, and everybody's got their own theory, right? When you have the kind of data sets, these kind of very powerful high dimensional data sets, there's no longer any debate, right? They're incredibly statistically powerful, and you've run pretty much every condition you could imagine running. And so you can't have someone coming along very helpfully saying, but did you try this? It's like, yes, we did. We've tried everything, and either it worked or it didn't work, but we can move on, right? We have a definitive outcome, and we can move forward with it. Anyway, so you hit back to the business model. So essentially it's software as a service, plus then a level of kind of consultation and partnership, really. We're working with those teams and those leaders who really understand that there's a fundamental shift that can happen with their experimental science, their experimental biology, and the data that come from that, and the insight that comes from that. And so, it's not an overnight change. It's something which has to be entered into very deliberately, but then it pays massive dividends, right? Because assay development, again, back to assay development, it's a critical path activity, right? Takes 9 months on average, and we can compress that timeline. So now, even if we did nothing else, then we've moved those drug discovery processes forward faster, right? And, the promises of AI is that then that keeps compressing even more, right?
Yes.
In future. But we're already, a good way forward with that. But then also we've been talking about assay quality, right? So we've made a better quality assay in a faster timeline. Yes. So now every time that assay is now producing data, and every decision that has to be made off the back of the data that's been produced by those assays are higher quality decisions. Those data sets, those screening data sets, the iterative medicinal chemistry that goes on, every time that assay is used, those scientists will be getting clearer insight than they would have done otherwise. And if we start training AI models on those data, those AI models will have clearer insight, right?
Absolutely.
And so, that is very exciting to both see, what's already possible, also where it could lead in future, right?
So I know a lot of these, the AI companies that focus on specific targets or platforms around that. But then, how you describe Synthace, this is very unique in that sense. Like, is this kind of a unique space, like in this design or so?
It is, yeah. Because what are we doing? We're combining, a kind of way of doing science, a kind of a theoretical framework, if you like, with software, with automation. It's kind of a mixture of expertise and software, and it's not a simple thing. And we've not the only reason we've got to this place ourselves is because we ourselves have been on quite an interesting journey as Synthace. We started out as a biology company, and we only made software to help us do these super powerful experiments, right? And so we are pretty unique, and that's both a real pro, because we don't have direct competitors, at least technologically, but it's also a con, because there isn't a pharma company out there with a line item which says, oh, we need to do automated multidimensional experimentation, right? So in startup world, this is called category creation, right? So you're educating the market about what's possible, and the problems that they're having that you can solve, and then
Mhm.
showing them how they can be solved. And that's a
Mhm.
much more kind of that's a much more sort of consultative process, and where you're chatting to people a lot more about science than you are about, like, the specifics of exactly how the software works, right? um,
Mhm.
yeah, it's a really interesting journey. It's a challenging journey, but for me, it's just like, I think you can tell what kind of a person you are often by what makes a good day for you, right? Like, when have you gone home happy? And the thing that never gets old for me is when I see the kind of sheer power of experiments that people are running in the lab, right? And when I get the chance to actually see those experimental data and to see the outcomes. Um, and that just, yeah, that's just really exciting. Really exciting to see.
Uh, Markus Gershater, Chief Scientific Officer and co-founder of Synthace. Markus, thanks for your time today.
It's been a real pleasure. Thanks for the chat.
Well, Amar, what did you think?
Yeah, it was a fascinating discussion. This is a pretty unique company focused in the design of experiments, and this is something that was usually when we're talking to a lot of the other companies, it's about they're developing specific algorithm or AI software to develop a platform for targets or a specific type of targets. This is a different type of company where they're using data analytics AI to get the best out of the experiments and even like to set up the right type of experiments.
One phrase he used that really stood out to me was, it's more about the data than the algorithms. I see that as a mantra and t-shirts. What did you think of that?
Absolutely, and we've talked that quite a bit, right? It's garbage in, garbage out, right? So if you don't have the right data, no matter how fancy your experiment, your algorithm is, it's not gonna be able to do anything because it's actually trained on. It needs to be trained on the right data. So that definitely is the mantra, right? It's data is the most important thing here.
He did talk a lot about the need to focus on data production and making it not only better but faster. Are people thinking about it in those terms with regard to their data collection, or are they just thinking about feeding the beast of AI?
I think that has started happening quite a bit. And I would say, especially in research, the scientists are very much aware of that. Because in science, these kind of experiments, especially the omics experiments, have been going on for a couple of decades now, more than actually almost 3 decades now. So the scientists in the pharma companies or in academia are used to setting up these kind of experiments and then making sure that all the data is collected. So I think that cultural transformation has happened in research. Not sure about commercial. In commercial, I think that in the pharma, I believe that's still happening at this point. Some companies are more ahead than the others. I see, like in the value chain of pharma, it is, I would say, research is where it's most advanced. Even in clinical development, when it's about clinical trials and running those, of course, the clinicians are very much aware of that. But when it comes to getting some new aspects, running some different new types of assays or so, the clinicians they need to be very well aware of that. So I would say culturally, it's there in research. In some of the other areas, it still needs to be it's systematized in the other areas.
He talked about enabling experimentation in a high dimensional and automated way to address the complexity of biology. How novel is what Synthace is doing?
See, this is something that needs to be done for the right assays, right? And we did talk about biological systems being very complex, having so many different factors. So for you to get the right conditions, you do need to play around with a lot of these conditions. And then that's what he talked about. This is the design of experiments, right? That's the field. So it needs to be done very systematic way. Um, I think it varies like with different scientists. Some scientists can be very disciplined about it. Some others may not be. So what they are bringing is they're just bringing like this. They're just and then they're making it like to be like very much a science, right? Like, okay, boom, boom, boom. You have to go through this, and you have to do these experiments. You have to try out all of these. So making sure, like, I mean, what they are enabling is making sure that we can that the scientists can do the best experiment that's possible. And that, to me, like having that kind of a disciplined approach, I believe is pretty interesting. It should be. I mean, that should be how all experiments need to be running in the ideal world, and they're trying to do that.
Marcus, at the end of the day, is talking about changing the way scientists in drug discovery have traditionally operated. He talks about being able to win them over with powerful data sets, but how big a change do you think he's advocating, and how much of a barrier is it to change the behavior of the way drug developers and scientists operate?
So it is going to be how disciplined the scientists are, right? I mean, if they really want to go through all the different conditions or so, I think those are easy to convert. The people who, but also at the same time, you have to see, right, that there are people who are more like who are more comprehensive and like more process oriented, versus there are people who are more like, okay, well, I think this is gonna work, and this is this, hey, I have done, I have a lot of experience in this area. I know what should be the ideal conditions. Let me just do it my way. I don't want to try out a lot of different things, right? So there are people who are more like, okay, well, I know what I'm doing. What is this gonna add? So I think those are the people who are going to be a more like challenging to convert into this, where this is really like going into systematic way, like and looking at the entire space, the multidimensional space that he talked about of the factors to really figure out. So it's going to depend on the personalities. As I say, like some people are right there, some people are not there.
Well, it was an engaging discussion, and I'm looking forward to the next one. Thanks.
Yes, absolutely. Thanks, Danny.
Thanks again to our sponsor, Agilisium Labs. Life Sciences DNA is a bi-monthly podcast produced by the Levine Media Group with production support from Fullview Media. Be sure to follow us on your preferred podcast platform. Music for this podcast is provided courtesy of the Jonah Levine Collective. We'd love to hear from you. Pop us a note at danny@levinemediagroup.com. For Life Sciences DNA and Dr. Amar Drawid, I'm Daniel Levine. Thanks for joining us.